The urinary system. The urinary system. The main function is that of an excretory system. It removes waste products from the body in the form of urine. It also has other functions. These are regulating water and sodium levels in the blood, converting the fat-soluble form of vitamin D to a water-soluble format, and the kidneys secrete a hormone called erythropoietin. It stimulates the production of red blood cells. The structure. The image to the left of the slide shows you the basic structure that would be found in the female and the male of the dog and cat. It is made up of 2 kidneys, 2 ureters, a bladder and a urethra. The urinary system. The number one function is to maintain the body's homeostasis. Homeostasis is the balance of all things in the body, so this is vitally important. The urinary system is responsible for removing waste products, including surplus water. The structures that make up the system are the 2 kidneys which filter the blood, the pair of ureters that carry the urine from the kidneys, the bladder which we class as a storage organ, and the urethra whose purpose is to carry urine out of the body. The kidneys are described as being retroperitoneal due to their position. The function of the kidneys is to excrete metabolic waste products and regulate the extracellular fluid. The 2 kidneys lie against the dorsal wall of the abdomen, and the right kidney is about half a kidney's length cranial to the left kidney. The indented part of the kidney is called the hilus and is the point at which the blood vessels, nerves and ureters enter or leave the kidney. On the X-ray we can see normal kidneys in their position, and the image also dictates to you where aspects of the urinary system are too. Here is an X-ray of the bladder in relation to a normal bladder X-ray. Top left. The bottom left is actually where we have used a contrast medium to actually bring up the bladder in that white format to be able to identify any issues in there. Cross-section of a kidney. There are 4 main areas. A capsule that is a protective layer of irregular and dense fibrous tissue. The outer cortex, which is darker red in colour. The slightly paler inner medulla, and the hollow connecting area of the renal pelvis, which contains white fibrous tissue and collects urine for passage out of the kidney. Functions of the kidneys: urine formation, the maintenance with other organs of the correct pH of the body, we term this osmoregulation, and this is all part of hemostasis. The conversion of vitamin D to an active form, and hormone production, and these hormones help to control blood pressure, make blood cells and maintain bones. The kidney nephron is considered as the functional unit of the kidney. Each kidney contains approximately 1 million nephrons in kidney tissue, and they are the mechanism by which the blood is filtered and urine is produced. And each is a long tubule that is divided into several parts. Nephron structure. The nephron starts in the cortex of the kidney with the Bowman's capsule, which contains the glomerulus. This is fed by an afferent arteriole and drained by an efferent arteriole, and together we term these structures as the renal corpuscle. The nephron structure. As the tubule leaves the capsule, it makes several twists and turns, and this part of the nephron is called the proximal convoluted tubule. The proximal convoluted tubule is lined with simple cuboidal or ciliated columnar epithelium on one side. The opposite side usually has a microvilli forming a brush border to increase surface area for reabsorption of electrolytes and water. The next part forms a long loop of Henle that dips down into the medulla, then returns to the cortex. The loop of Henle is a U-shaped tube that leads from the proximal convoluted tubule. It is located in the renal medulla. It has thin, simple squamous epithelia on the descending loop, which is thicker than on the ascending loop, and this filters useful products out of the kidney. There is then another series of convolutions, shorter than the proximal convoluting tubule, and these are termed the distal convoluted tubules, before the nephron empties into a collecting duct that serves a number of nephrons and carries the urine through the medulla to the renal pelvis. The distal convoluted tubule is lined with cuboidal epithelial cells, therefore it does not have a brush border. The collecting ducts that are lined with columnar epithelial all discharge their contents into the renal pelvis at the apex of the pyramids of the medulla. Here is an image depicting the different parts of the nephron. Kidneys filter blood. They produce a filtrate that goes through modifications within the renal tubules to produce urine. Urine is very different to its initial composition, and also so is its volume compared to that of the initial form of said filtrate. For every 100 litres of fluid filtered from the blood, only 1 litre is produced as urine, and 99% of the original filtrate is absorbed back into the blood. Blood starts its journey via entering the kidney and is carried to the capillaries that form the glomeruli. Glomerular function. High blood pressure. The hormone renin has an effect on this area. High pressure causes the forcing of fluids and small molecules out of the blood to move through the pores of the basement membrane and to move into the capsular lumen. Larger sized molecules, i.e. red blood cells, plasma proteins, as substances that are bound to protein molecules such as hormones, are retained in the blood. We term this ultrafiltration, and in turn we call the filtrate glomerular filtrate. This filtrate is very dilute, containing only 1% chemical solutes, with the other 99% being that of water. The loop of Henle regulates the concentration and also the volume of the urine, and this will be dependent on the status of the extracellular fluid. The filtrate firstly flows into the descending loop, then onwards into the ascending. Within the descending loop, the walls are permeable to water but cannot absorb sodium. Water is drawn out of the tubule via osmosis and is pulled by the concentration of sodium in the surrounding tissue. Thus the filtrate becomes increasingly concentrated as it passes down the loop, reaching maximum concentration at the tip. Within the ascending loop, walls here are impermeable to water but contain sodium pumps that can actively reabsorb sodium into the tissue and capillaries. So within the distal convoluted tubule we have final adjustments being made to the chemical makeup of the urine due to the status of the ECF, extracellular fluid. We've said we have reabsorption of sodium, smaller amounts than in the proximal convoluting tubule, and reabsorption of water by osmosis will vary and is controlled by the hormone aldosterone. Secretion of potassium secreted in urine to replace the sodium ions. The acid-base balance is regulated by the excretion of hydrogen, and remember blood has a normal pH of 7.4. If pH falls, the kidney will in turn excrete hydrogen ions into the urine via the distal convoluted tubule, thus returning the pH to normal. If the pH were to rise, the kidney will in turn stop excreting such ions to keep them in the blood, thus returning the pH to normal. This is all part of the process of the balancing and homeostasis of the body. The collecting duct. Final adjustments to the volume of urine due to that ECF status. The antidiuretic hormone known as ADH can cause change to the permeability of the walls of the duct to the water. If the animal is dehydrated, then the volume of plasma and ECF will be reduced. Thus, ADH will be produced to increase the permeability of the walls of the duct to water. This will then ensure that water will be drawn through the walls by the high sodium ion concentration and into the plasma and ECF. Due to this process, the ultrafiltrate that started its journey has now been concentrated, and thus it becomes concentrated urine due to repeated absorption processes. The ureters. The ureters, we have one from each kidney, are thick-walled, narrow tubes that carry urine from the renal pelvis to the bladder. They're lined with mucous membrane and have smooth muscle which undergo peristaltic movements to move the urine on down towards the bladder. The ureters enter the bladder close to its neck, and there is a single flap that prevents the return of urine up the ureter under the high pressure of a full bladder. The bladder is considered to be retroperitoneal. It moves within the pelvic area dependent on how full it is. Each ureter enters the bladder through an area known as the trigone, which acts as a muscular valve. The bladder is capable of considerable distension, as it is smooth muscle. The lateral ligament holds the bladder to the lateral pelvic wall. The bladder is lined with transitional epithelium, which continues into the urethra. The lining of the empty bladder becomes highly folded, allowing it to have massive expansion when it is full. The neck of the bladder ends in the bladder sphincter, which controls the flow of urine out of the bladder into the urethra. It has two concentric parts: an internal sphincter, which is smooth muscle under involuntary control, and an external sphincter, which is striated muscle under voluntary control. The urethra. In the male, the urethra carries urine from the bladder to outside the body. It is divided into two parts: the pelvic urethra and the penile urethra. In the female, it carries the urine from the bladder to the external urethral orifice, which opens into the genital tract. There is a ridge known as the urethral tubercle, and this is helpful for actually catheterising the bitch when required for urinary catheterisation. Here we can see the differentiation of the urinary system based on the animal's sex. So here we have a male and a female. The male urethral species differences. The dog has 3 openings close to the neck of the bladder, 1 for the prostate gland and 2 for each of the deferent ducts. The vas deferens, 1 for each testes. The urethra runs through the pelvic cavity, over the ischial arch of the pelvis to join the erectile tissue. The penile tissue continues until reaching the tip of the penis. Male urethral species differences, this time in the cat. The cat has a short urethra cranial to the opening to the prostate glands and deferent ducts known as the preprostatic urethra. This is not found in the dog. The cat has no penile urethra outside of the pelvic cavity. Close to the end of the urethra are paired bulbourethral glands, which we've talked about in your reproductive lesson. The urethra opens to the outside in the perineal region, ventral to the anus. Micturition is the technical term used to describe the action of expelling urine, usually a reflex but can be overridden by the brain. There are stages. The bladder becomes distended with urine. The stretch receptors within smooth muscles in the bladder send nervous impulses to the spinal cord. The impulses are initiated to produce muscle contraction and relaxation of sphincters. The urine is expelled.